My title is Metabolic and Epigenetic Alterations During Aging, and I'm pretty much going to stay to that. That was my original title, and that is more or less the topic we're going to cover. Can everyone hear me okay in the back? Okay, excellent. So I'm going to try to self-regulate. See how that goes. So what I want to tell you about this afternoon is really broken down into two areas that I'm going to spend time on. The first is I want to cover some background and research overview of our group, as well as some work that the Wisconsin Institute for Discovery is initiating, and we need your help to solve. So there'll be a call out for crowdsourcing some of these efforts. And in the second half of my presentation, I'm going to dig a little bit deeper and talk about a pretty long-term project that we've been working on. If you're studying aging, you do have to wait a while for things to happen. And so we're starting a mouse model for aging and the effects of caloric restriction in a diet and a genetic background that I think is proving to be really interesting and totally unexpected. I think now there's a pretty good appreciation for the role of non-genetic pathways, non-genetic mechanisms to control disease initiation and disease development, as well as altering lifespan. And an area that I'm particularly interested in is understanding epigenetics, understanding the epigenome. What is this information? How do we harness it for good? Okay. Two really good examples for epigenetic phenomena come from sort of a model organism and an unfortunate incident during World War II called the Dutch famine, where children born to mothers that experienced the famine in the winter of 44 and 45 at the end of the war, those children that were born to mothers who experienced the famine during the first and second trimester were way more likely to get metabolic disease, diabetes and obesity. Some work more recently has suggested that, in fact, there is an epigenetic association with those individuals that have dysregulated DNA methylation, which is one of the mechanisms we and others think is an important epigenetic mechanism. The other example comes from the agouti allele in mouse, this particular strain of mice that's very susceptible to DNA methylation around a locus that determines satiety as well as coat color. So these mice are genetically identical, but one was exposed, for instance, to bisphenol. There's many ways you can alter this allele and alter the methylation. So you're not changing the DNA sequence at all. You're just changing the methylation content. And one can do that prenatally or in utero with folate treatment. You can alter the methylation status, or exposing the pregnant mothers with bisphenol A. Bisphenol A is a compound that's used in producing plastics. But these compounds can basically alter this epi allele, resulting in a yellow obese mouse versus, again, a genetically identical lean brown mouse. Okay, so these are epigenetic phenomena. My group and actually several others on this campus have been quite interested in the effects of caloric restriction on parameters of health. We still are trying to understand the epigenetic component to this, but it is a very profound way of altering health in almost every organism that's ever been attempted, which includes yeast, worms, mice, rats, non-human primates. And a recent study that just came out, I read it on CNN last night. I wasn't able to read the the article, the research article, but basically there's a study that came out, 15% reduction in calories in humans. It doesn't necessarily extend lifespan, but parameters of health include the way our bodies handle glucose, for instance. So reducing metabolic syndrome parameters or other aging parameters. So that sounds pretty cool. But there are advantages to studying this in a model organism. And later today, well, not that much later today, I will be telling you in more detail about our studies in mice. But largely, these are epigenetic phenomena. So what do we mean when we talk about epigenetics? We're all familiar with the flow of genetically encoded information shown here. This is sort of the central dogma of biology. It's an oversimplification for many of you, as you know. It's more complicated than this, but essentially DNA encodes information to DNA is transcribed to RNA. RNA is then translated to protein. Protein, enzymes, and other proteins perform molecular functions which can lead to observable phenotypes. Now, we generally believe that epigenetic information is impinging at this step, the decision of which genes to express. Okay? And I like to think about it as a computer, as an analogy to computer hardware and software, where the genome is really the hardware. There's not much you can do about that, although there are improvements to potentially genetic therapy. We're still a bit of a ways off there. But one can think about the software on an instrument, on a computer, as the epigenome. So a different application really creates a new functional entity on your cell phone, or so those of you, this is a computer. Those of you that, see, there's a lot of youngsters in here, they probably don't have computers anymore. They just have a phone. Okay, and software are now apps, so that's why I had to clarify that. Okay, so let's talk a little bit more about this information. What does this information look like? And again, this is a quick summary of epigenetic information. We think about epigenetic information that sits on top of the genome. There's a couple ways that manifests. One is actual chemical modifications to DNA, as well as chemical modifications here shown as lollipops on proteins, on this protein sequence. These are things such as phosphorylation, methylation, acetylation, and so forth. This occurs on histone proteins, and histone proteins, you can think about them as the spools of DNA. They're the spoolers of DNA. So they wrap DNA up, and they can be, the accessibility of the DNA is regulated in part both by modifications to DNA, as well as modifications to the histones that wrap DNA. Okay? This is a source of epigenetic information. Now, what's really cool about this is that these modifications are catalyzed by enzymes, okay, protein products, that in fact utilize core metabolites from intermediary metabolism. Okay, so this is sort of the TC. Can you see my pointer? Can you see it okay in the back? Okay, I'm barely seeing it, so hopefully you know what I'm pointing at. This is the TCA cycle here, and some of those metabolites can drive, help the enzymes that remove methyl groups from DNA or histones, and then of course acetylmethionine, which is a key metabolite that drives the methylation reactions and so forth. And acetylation is driven by acetyl-CoA, central to life. All these are central to life. But it's interesting that the enzymes that modify chromatin, modify these epigenetic states, require these metabolites. Okay, so this opens up the possibility, right, that you can alter the epigenetic state through diet, through other forms of metabolism. Okay, and in fact, some work that we're quite interested in continuing to pursue is the role of the gut microbiome in producing metabolites producing metabolites that are vitamins that are essential for humans, or producing metabolites that are not so much essential but can modulate the epigenome of the host. Things like these short-chain fatty acids. So fairly recently, a couple of years ago, we collaborated with Federico Rey, a bacteriologist here on campus, to study the effects of the microbiome on the epigenome of the host. This work from my group was primarily done by Kim Krautkramer, an MD PhD student from here. And what we determined is, in fact, diet matters, and the fact whether or not the mice are colonized. So we looked at germ-free versus colonized, and under two different dietary conditions. The bottom line is that there were very clear epigenetic differences and at the level of modifications to chromatin, and transcriptional differences that are due to the diet and the microbiome. So we're interested in what are the factors that affect the epigenome. So diet, lifestyle, and stress, for instance. How can this lead to alterations in this epigenetic information with a clear emphasis on understanding how the gut microbiome influences the epigenetic states within the host. Okay. What's the molecular mechanism? So we're interested in what these factors are, but we're interested in what the molecular mechanism behind this effect is. So we'll measure, for instance, how chromatin is affected through gut microbiota, diet, lifestyle, where you live. These are all factors that we're thinking about. And when I mean we, I mean our group, but also the greater collaborative effort here on this campus, which I'll tell you about shortly. We're very interested in how these affect gene expression patterns, how this can lead to dysregulation in different tissues, particularly the brain, where we're interested in exploring these human afflictions. I'll just give you a quick example, because Alzheimer's disease is, in fact, one of these human conditions we'd like to understand. And we're lucky in my group to have a scientist, James Dowell, who's been studying this, who's been trying to establish a link between AD, so mouse models of AD, and altered chromatin states. And what James has found is, in fact, there's some pretty profound changes in two different models of Alzheimer's disease, two different mouse models of Alzheimer's disease. So what James has found using a mass spectrometer, a proteomics approach, that there's two groups of peptides. So these are modified peptides within the cerebellum and hippocampus. Actually, they're only significant in the hippocampus, which is pretty interesting in these AD models. Not significant in the cerebellum. But James has found that, in fact, there's a group of peptides that showed altered regulation. This peptide here, H3K9, K14, and this peptide K27 and 36, these two lysines that are really major targets of modification in eukaryotes. So they are dysregulated in the PS1 mouse model. And perhaps even more amazing is that when James looked at the amount of histone H3.3. So this is a histone that's really important for post-mitotic cells. Okay, so this would be obviously very crucial for the brain. And what he saw is that there is a loss of soluble forms of this histone H3.3. This is a critical histone for gene expression. And why is it critical? Well, it's one of these histones that gets replaced during transcription and during real active, dynamic regions in the chromatin. And it's the only one that's being expressed in post-mitotic cells. And what what this data suggests, in fact, there's a dysregulation of these modifications we think through the inability with the inability to evict those histones, or the inability to deposit new ones onto this. So what happens by having a low pool of histones to call on, to replace during turnover, we think we're getting in a stuck state of chromatin. We're in a epigenetically stuck state that basically produces a very fixed or a transcriptional state, perhaps leading to neuroinflammation. There's some data in an analogous astrocyte model that suggests that this phenomena may lead to a response, a transcriptional response for neuroinflammation. So we're excited about this data, and James is extending this work into humans. He obtained an ADRC pilot grant to investigate these alterations in chromatin and these dynamics in human AD samples. We're very excited about following up on that. Okay. Oh, I'm doing just fine with time. In fact, I think I'll take a... Can I take a quick break? This is a transition time. Okay. Thank you. So I'm an investigator in the Wisconsin Institute for Discovery, and in fact, I've been here now for maybe seven or so years, and I wrote a proposal when there was a call for proposals on campus to move your lab into the WID. It was a competitive proposal, and you could hire new faculty to campus. I mean, how often does this happen? Not that often. I guess it's happening now with the cluster hires, but this doesn't happen that often, right? There's not this kind of opportunity. I thought this was a wonderful opportunity to bring new investigators to campus, and we developed an epigenetics theme here in the Wisconsin Institute for Discovery, which we're now calling a hub because we've basically created a physical and intellectual entity that is bridging to the rest of campus, and that we're super excited about, and creating a community there. But we continue to look beyond our group as a single hub. And as you know, we're under new management. Joe Handelsman is our new director of the Wisconsin Institute for Discovery. That's where you guys are right now. Well, it's complicated. It's the Morgridge Institute for Research, and it's the Wisconsin Institute for Discovery, and you are in the Discovery building. So WRF kind of owns this part. So technically we're in WRF space. Okay, but Joe, we're really writing chapter 2 for the Wisconsin Institute for Discovery, and both Joe and I feel that really this place, as I think it was always intended to be, is a catalyst for interdisciplinary work. Okay, as you know, we have engineers, we have data science folks, biochemists, molecular biologists, engineers, and so forth. So, did I say engineers? Did I say that twice? What does that mean? What is that? I always have that problem, so I'm not sure. Maybe, yeah, okay. Anyhow, so one of the things that we've been thinking about is getting a group of people together and trying to solve a big problem, sort of a grand challenge. And I just want to briefly tell you about that, because it's one we're very excited about. It's one I've been working on quite a bit with my fellow colleagues, both here in this building and on campus. And it's very pertinent to this group. We'd like to think we could create healthy minds for a lifetime. So we're calling this goal BrainSpan 100, with the goal of maintaining healthy brain function throughout 100-year lifespans, right? I mean, lifespans are increasing, right? As a society, we're getting older. We're living longer. But what's the point of living longer if you're suffering from dementia, if you're suffering from anxiety, if you're suffering from depression, and so forth, all right? So can we imagine a society where we have healthy minds and healthy bodies for 100 years? So WID and our network of colleagues are trying to build a research pipeline that permits the integration of molecular and clinical data with a major focus on the factors that affect brain function and go beyond the genome. I think the genome is great. I love the genome, right? But the genome is so last 20 years. No, it's not really, but I do think when we study individuals, when we study groups, knowing their genetics, yes, we should know that. We should be able to do whole genome sequencing on everybody. That would be awesome. Okay, that's a starting point. But what else do we need to know about them? Okay, what else do we need to know about their epigenome to effectively understand their disease, their susceptibility to disease? Okay, so in our efforts, we really want to focus on the microbiome and the epigenome because we have real strengths on this campus in those areas. And so we're envisioning a pipeline, building a platform, I guess one could call it, and where we can feed in a number of important biological questions. The goal is to funnel those questions into a multi-omics platform. So where we would be performing microbiome analysis, genome analysis, hopefully whole genome, but that's super expensive. So maybe just SNPs, maybe exome sequencing. Again, depending on the study, depending on the amount of funds available. So, and the epigenome analysis, what does this mean? Well, not just methylation analysis. And again, this is why I think Wisconsin has real strengths in this arena. We can do a number of epigenomic analyses, including the histone profiling that I showed you with James's project. Chromatin accessibility, transcriptome analysis, of course, proteomic analysis and metabolomic analysis, in addition to known clinical parameters. So the idea would be to funnel in. I guess I probably don't have time to tell. Well, no, I probably don't have time to tell you about how some of these projects, who on campus is helping us in these areas. Again, if there's time at the end, I'll take you through some of our colleagues on campus that are helping us and bringing us very unique cohorts to study in this regard. But again, the idea is to build a platform that allows this multi-omic analysis, again, including like electronic health records, for instance, other clinical epidemiological data. The idea then, the output would be potentially diagnostics. Can we predict health outcomes? I think we also have a real strength on this campus in data science. And so one could envision new data science platforms that come out of this. Again, a real strength of Wisconsin, we have many here. One is the ability to understand mechanism. Okay? Some of these are simply discovery tools. Okay? And what we want to be able to do, come up with a really cool hypothesis, maybe a handful of really cool hypotheses from this data, and say, you know what, we can test that. We can actually go in, provide cause and effect. Wouldn't that be cool? So we're generating, one could envision generating genetic models using, if we're focusing on the microbiome, using our notobiotic facility here, define communities compared with germ-free mice, for instance. And we're in the process of developing and engineering organoids to test very specific questions about the link between the microbiome, for instance, in the gut and brain function, using potentially a co-culture of organoids of bacteria with an intestinal organoid connected to a brain organoid. I know it's kind of science-y fiction stuff, but this is what we're talking about right now, and excited to build that out with all the help we can get from folks here at Wisconsin. Okay, so with the remaining time that I have, I do want to take a slightly deeper dive into the effects of caloric restriction. And the question I brought this up before about the improved health parameters under caloric restriction, lower cardiovascular disease, lower dementia, lower diabetes, heart disease, and so forth. So what's the molecular basis for this? This is something we're very keen on. 10 minutes, holy crud. Okay. Thank you for that. Okay. So the question is, how does caloric restriction, and this enzyme in particular, this is a mitochondrial enzyme, SIRT2 and 3, and I'll tell you why we focused on SIRT3, okay? How does it impact mitochondrial function with age, and are these effects on health span or lifespan? The reason we're focusing on SIRT3 is we previously, in collaboration with Tom Prola, demonstrated that SIRT3 was critical for the prevention of age-related hearing loss under caloric restriction. So it turns out that black 6 mice lose their hearing at about 12 months of age, but you can prevent that with caloric restriction. But you can't if they have no working copy of SIRT2 and 3, okay, which is a mitochondrial protein shown here deacetylating this metabolic enzyme. I'll skip that and just go to the fact that SIRT3 is upregulated. It's a mitochondrial protein. We think it's the major protein deacetylase, so it removes acetyl groups from the side chains of proteins, lysine residues. And for all intents and purposes, we see that most of the, when it deacetylates proteins in the mitochondria, and most of these are metabolic proteins, it increases their activity. So acetylation typically reduces activity of these enzymes. Okay? SIRT3 deacetylates, activates the enzyme. So here's the experimental design of the study. This was all carried out, most of what I'm going to tell you about was carried out by a postdoctoral fellow in the lab, Rush Dillon, in collaboration with Tom Prola. So basically we took a wild-type mouse, control diet, versus a calorically restricted diet, so about 25-30% lower calories without malnutrition, okay? Wild type, and then the knockout, notice the little circle, that means the knockout. These are all artwork, this is all artwork. I'm sure most of what I'm showing is artwork from Rush. He's very talented in the lab and in drawing, obviously. Okay, so we took animals 5, 15, and 25 months of age, and we fairly focused on mitochondrial respiration, okay? So as you know, there's all of these cool complexes that sit in the inner membrane of mitochondria. They pump protons into this inner mitochondrial space, and in the process, they extract electrons from the stuff we eat, okay? And ultimately, they build up this electrochemical gradient, use that to drive the synthesis of ATP, okay? This is where most of our ATP is coming from. These set of reactions right here. Okay, I apologize. These things jumped around in this conversion from Mac to PC. You know what? I'm running out of time. I'm not going to show you data. It's 3:30-ish? Okay. You guys probably want coffee. So I'm going to skip through this. Rush, I'm really sorry. Are you in the audience? Yeah, sorry. And he doesn't have a poster. You don't have a poster today, do you? Ah, okay. All right. So I'm going to skip through this, and I'm just going to tell you what I would have had I gone through the data. So mitochondrial aging is nonlinear in most tissues. You probably could have seen that from the distribution of those bar graphs, which is really wild because most people, when they study aging, it's like, let's take an old and a young group. Well, we took old and young and middle age. And turns out some of the parameters go like this, like mitochondrial respiration sort of looks like this. Some it's decreasing sort of more normally, but it's nonlinear, most tissues. At the oldest age, SIRT3 is required to maintain high coupled respiration in the brain, heart, and liver. So SIRT3 is critical for the highest coupled respiration in these animals at the oldest age, okay? How many minutes do I have? 7 minutes. Oh, well, maybe I should go back and show you all the data. No, I won't. You remember, so 1970, remember Sesame Street? Which one of these things is not like the other? It's a real narrow window. People here probably know what that means. But I remember vividly. It's like, you're looking at things, you're looking for where's the difference here? Oh, sorry, I have to go back. Okay. Sorry, I got to be tethered. Sesame Street, right? Which one of these things is not like the other? And that came out in 1970. I looked that up just now, that song. There's actually a song, and I won't sing it. But the point is that in middle age, okay, what is this? This is actually percentage of respiration. So oxygen consumption as a function of either coupled respiration, here, uncoupled reserve, that means what you could do if it were maximum ATP generation, and then leak. So this is basically oxygen consumption that's not driven by ATP synthesis, okay? So which one of these things is not like the other? 15 months of age doesn't look like 5 or 25. So another way to say that is 5 and 25 look like each other in terms of the amount of uncoupled respiration and leak. So this is the least, so this area up here, you can see there's the most coupled respiration as a function of the total respiration, which is wild. What does that mean? Okay, quick summary. Middle-aged mice show the highest coupled respiration as a percentage of all oxygen consumption. 5 and 25 mice look more like each other than they do to the 15-month-old mice. And except for gastroc muscle, caloric restriction and SIRT3 are required at old age to maintain the coupled respiration characteristics seen with middle age. Again, it's probably too late in the day to take you through these cool bar graphs. And it's certainly too late in the day to take you through this one. But this is just to illustrate all of the things we've measured in these mice, all of the parameters we've measured. That includes mitochondrial DNA content, Vmax of each of the electron transport chain proteins, their protein levels, the specific activity of all those 5 complexes. Okay, that's just a handful. So it turns out that mitochondrial DNA copy number is not a significant factor, and that's consistent with the EM electron data density that I didn't show. Complex 1, specific activity tends to decrease with age and is likely due to decreased subunit levels. Okay. But this is quite interesting here. A calorically restricted wild-type mouse shows higher complex 2 specific activity in old heart, liver, and brain is higher than any other age. So you can see here, this is the wild-type CR animal, has really high coupled respiration. Okay, what does that mean? So in the last few minutes, let's talk about lifespan and health span. Okay. So it turns out we had a couple exercise physiologists join us last summer from Brazil, and they carried out these experiments. Where they are experts in exercise physiology, and they measured critical speed in these animals. Critical speed is essentially, it's a really cool parameter, but it's essentially a measure of whole animal aerobic fitness. And if I just want to draw your attention to the red guys here, the red bars. The red bars are middle-aged mice, so 15-month-olds, because we have data on all 4 categories. The only one that's significantly different than the other is the wild-type mouse under CR. Actually has the highest aerobic fitness, the highest aerobic capacity. So that suggests that the ability of CR to improve caloric restriction to improve critical speed, so this measure of overall aerobic capacity, requires SIRT3. And SIRT3 deficient animals show impaired critical speed at old age. Okay, but what about 2 minutes? Oh, this is perfect. It's perfect. Thank you for being here. and keeping me on track. This is another crazy moment, okay? Not that the last ones were crazy. That was a bit more expected. This was unexpected. There was an unexpected and unexpected part of this. And that is, people already knew that mice live longer under caloric restriction, as we've defined it. And that's this data right here. This is the CR effect. So average lifespan is increased under caloric restriction, regardless of whether these mice have a functional copy of SIRT3 or not. Okay? But something really crazy happened at really old age, or I should say, in conditions where we would calculate maximum lifespan. So average lifespan, not so different between this group and this group. Sorry, these two and these two. Okay, CR. This is the CR effect. What I'm talking about here is maximum lifespan. So the last, say, 10 to 20% of those living, what is their average lifespan? And it turns out that if the mice have, if they don't have SIRT3, they live longer. They have an increased maximum lifespan. That trend, although not statistically significant, was also seen in the control animals, the control diet. So these animals, the knockout animals, are actually living longer than caloric restriction, which I'm not aware of any other treatment that extends lifespan beyond caloric restriction, at least in this animal model. So this blew us away. It was exact opposite of what we thought. So what's happening? I don't have time to tell you what I think is happening with that data slide. And I'm sorry, Alexis and Amy, but this is the last slide. Data slide. Actually, this is a summary slide. So what we think is happening is that lower mitochondrial energy capacity is associated with increased lifespan, but lower whole body aerobic fitness. Okay, so Rush and I came up with this. We're trying to write up this paper right now, and this helped us sort of conceptualize how to think about this. So think about it as the capacity, the overall mitochondrial oxidative capacity of the mitochondria, okay, from high capacity to low. If you have low capacity, so consider that your health span, right? The mice don't do well here. They need higher capacity. So you need to be SIRT3 positive, okay? But that doesn't seem to matter for lifespan. The demand is not there. So you can imagine you can have low mitochondrial capacity with increased lifespan. That's what this data is suggesting. It suggests that health span and lifespan, this is kind of crazy to think about. Health span and lifespan may not always be connected. Anyway, food for thought. Okay. This is, actually I lied. This is my last slide, but this is the most important slide. I tried to indicate the folks in my group who have been involved in this work, and I tried to mention some of our collaborators in passing. Unfortunately, I didn't get a chance to talk about the extent of our collaborators on these newer projects, but just within the aging project, I want to thank Tom Prola, my longtime collaborator on these aging studies, and two of his scientists, Paul Van Ginkel and Vivian Fu, as well as our two Brazilians who came in and did the awesome job on the, Claudio and Fulvia, on the exercise physiology analysis. So with that, I'll stop, and hopefully there's time for questions. Thanks a lot. Okay. Yes.
So actually, I have a question for both speakers. So earlier, Dr. Fedorov talked about nutraceuticals as one solution to the phenomena that he's seeing. And your work and work of others on this campus have really focused on caloric restriction. So knowing humans, Caloric restriction is a major problem. In fact, I think on the front page of the New York Times or the Washington Post today, one of the two, there's an article about the dramatic increase in obesity, which suggests that people are not food restricting. So if we're going to choose a practical strategy for increasing not only lifespan, but health span and cognitive health, which is the better one?
I'll just say a quick note on the caloric restriction. You're right. I mean, so 30% calories. So if you're trying to mimic the studies that, again, I failed to mention Rick Weindrich, who's famous for his work and his colleagues here at Wisconsin studying the rhesus monkeys and longevity, and that's about a 30% reduction in calories. The same is true for the mice model that we've been working on, and apparently that, and this paper I referred to, nobody can do that. Well, I shouldn't say nobody. Very few people can do 30% reduction, but in this newer study that was just published in Cell Metabolism, again, I just read about it on CNN last night late. That was the press release. People weren't restricted to doing 30%. Basically, they just simply measured how many calories were they restricted, and some people could do 15% quite effectively, and saw some of the improvements in terms of metabolic parameters. Now, one of the reasons we study caloric restriction is not to promote. There's no way I would ever do caloric restriction. I don't want to live to be 100 and miserable every day of my life. So I think what we're trying to understand is a molecular basis for this, so that we can, perhaps through drugs or through maybe an intermittent fasting, something that's tolerable by humans, not 30% caloric restriction. So we want to understand at a molecular basis.
Well, first I would say we want to promote quality of life. And if quality of life is deteriorated, and I've met a couple of people who are doing their own caloric restriction work, including some who work for the National Institute of Aging, they look terrible, and I don't think they feel terribly good about how they look. So their quality of life, by definition, is probably suboptimal. Is that going to be true for everyone? I don't know and wouldn't say or speculate on. I think preserving health span and quality of life seemingly would be the right endpoints. And although the data that you reviewed and that which we didn't see would suggest that turning down mitochondria seems to promote lifespan but not health span, most of us really would like to be able to be vital and engaged, and having the capacity to do more aerobic work and be able to respire, mitochondrially speaking, I think would be associated with a better quality of life. So at least for me, I'm voting for that.
Can I stimulate it? Can I just throw something out there to this audience? And so with this idea of the brain span 100, of this audacious goal, right? What we're looking for is community involvement. And I mean community, I mean other scientists, I mean coming from places we don't even know. I think this is an effective strategy to think about solving a really important problem in society. It's not going to be a handful of scientists. They're going to help, okay? But we need help from the public. We need help from areas of science that haven't been asked to help, right? And so I'm asking to help. I'm asking for your help, right? And people say, oh yeah, I just heard this talk. This is what they're trying to accomplish at Wisconsin, at the Wisconsin Institute for Discovery. Here's this audacious goal. How could we contribute in a way that we wouldn't have anticipated? That's where this crowdsourcing idea comes from. And I think, I don't think we think we're so smart that we can solve this problem on our own. And solutions to these problems are going to come from places we didn't expect. So I'm opening this up, okay, to the community. to help solve this problem. That can come in the form of course, ideas, time, money is always good. But anyway, but we don't know what those answers might be. So we need folks like you with your own individual expertise and your own network to help solve these kinds of problems.
So we're running ahead of schedule. The food is here, so I'm going to invite you to have something to eat, something to drink, and then visit the posters. We have, I believe, 42 posters representing a span of work from community engaged work up to basic laboratory science. So you can go from the south to the community and back again. Invite you to visit with our poster presenters, and we'll come back here. We have very generous award for poster presenters from the Hispanic family. Fun night Sunday set up in memory of the parents, and then we'll have a couple of closing remarks. So, your talk was Nicole. That's one of the reasons why we put it in. Thank you.
Sweet.